Thank you for taking the time to explore the sketchy biochem universe. I'm your trusty narrator, Ricky J., and you've been assigned to obtain surveillance against the Nitro Gang, who you suspect is running a multi-million-dollar illegal drug empire and pyramid scheme. Those don't seem to be on the same level, but here we are. You're conducting a stakeout inside a highly sophisticated van called the Nucleus. We'll begin this first chapter talking about DNA and chromatin. DNA is the magical blueprint that contains all genetic information inside cells. DNA, in the form of chromatin, is found in the nucleus of eukaryotic cells. DNA is also present in the mitochondria, but exists in the circular form and is only passed on by moms. Thanks, Mom. Two DNA strands come together to form a double helix. We'll represent this stakeout van as the nucleus of a eukaryotic cell. DNA will be represented as blue double helical wires that can be seen inside the van. Let's also mention RNA, which is found inside the nucleus and outside the nucleus in the cytosol. You'll see some single orange wires inside the nucleus. These wires symbolize RNA, which is similarly single-stranded. RNA is usually in the form of messenger RNA, which is an intermediate between DNA and proteins. RNA also exists as ribosomal RNA, transfer RNA, small nuclear RNA, and non-coding RNA. But we'll talk about that in another sketch. RNA contains a ribose instead of a deoxyribose sugar, which is found in DNA. Let's examine this map, which looks like a simplified version of a nucleotide. The Nitro Gang operates inside the nitro base, which is associated with a sugar house. There's also a helipad that connects to the sugar house, naturally. DNA and RNA contain many nucleotides. Each nucleotide consists of a nitrogenous base, a deoxyribose or ribose 5-carbon sugar, and a phosphate group. This differs from nucleosides, which only have a nitrogenous base and sugar. The pentagon-shaped sugar house is labeled from 1 to 5. These are the access points to get into the building. The sugar house is a deoxyribose or ribose sugar, and the helipad that attaches at point 5 represents a phosphate group. You'll see 2 more sugar houses to the right. Our Nitro Gang members reside there. The deoxy house has an H at point 2, while the oxy house has an OH instead. This is because deoxyribose sugars have a hydrogen at the 2' position, while ribose sugars have a hydroxyl group instead. Now it's time to meet the Nitro Gang, which consists of 5 members: adenine, guanine, cytosine, thymine, and uracil. Adele, Greg, Cyrus, Timothy, and Ursula. Adele and Greg are wanted for drug purification. The word purification sounds a lot like purine. Adele and Greg are wearing gold-colored sunglasses, which look like a 2-ringed purine. Purines are made from scratch or from the purine salvage pathway. The amino acids glycine, aspartate, and glutamine are needed to make purines. You can learn more by watching our purine sketch. Cyrus, Timothy, and Ursula, on the other hand, are wanted for running pyramid schemes. And pyramid sounds a lot like pyrimidine. Cyrus, Timothy, and Ursula, like cytosine, thymine, and uracil, are pyrimidines. Each of these hoodlums wears a hexagon eye patch, which looks like a 1-ring pyrimidine. Timothy is circled in blue and Ursula is circled in orange because thymine is only found in DNA and uracil is only found in RNA. These fancy handcuffs have a 5 and 3 end. They represent nucleotides held together by phosphodiester bonds. The 5' phosphate group of one nucleotide binds to the 3' hydroxyl of another nucleotide to make a phosphodiester bond. And if you don't remember any of this, it's okay, because you won't forget that lovely diester love note. You'll see another pair of handcuffs, but this time it's oriented in the opposite direction from the first pair. DNA strands are anti-parallel to each other, just like these handcuffs. Remember that 2 DNA strands form a double helix. The phosphate and sugar groups make up the backbone, and hydrogen bonds hold the bases together. In DNA, 2 hydrogen bonds bind adenine and thymine, and in RNA, 2 hydrogen bonds bind adenine and uracil. If you look back at our mugshot pictures, note 2 threads connecting Adele and Timothy and Adele and Ursula. You'll also see 3 threads connecting Greg and Cyrus. Kinda like how 3 hydrogen bonds link guanine and cytosine. Just as we suspect, 3 hydrogen bonds are stronger than 2, and it would require a higher melting temperature to separate guanine and cytosine. Maybe Greg and Cyrus are better friends than Adele and Tim? Check out the music coming out of that radio. Let's listen. Yeah, that's literally what the major groove sounds like. The big musical notes represent the major groove, which form when the DNA backbones are farthest from each other. Transcription factors and other proteins bind to the major groove to regulate transcription and DNA replication. And if you weren't blown away by that major jam, here's something minor. Yeah, those small notes are part of the minor groove, which makes pretty great stakeout music. The minor groove forms when the backbones are closest to each other. Let's switch gears and look at these dangerous grenades that just happen to be sitting behind the driver's seat. I'm glad I'm not driving. These minus charge grenades are just like minus charge DNA, but more dangerous. DNA has a net negative charge because of the minus charges on the phosphate groups. Let's say you decide to extract DNA from one of your cells for funsies. If you were to stretch that thread of DNA out onto a straight line, it would measure 6 feet, or 2 meters. It would suck trying to make copies of that. That's why DNA is packaged into chromosomes. During mitosis, DNA condenses and wraps around histone proteins like beads on a string. You'll see video footage of a tied-up, toned man. His tone, he's toned. Real toned. Note the blue wire wrapped around him. 2 subunits of H2A, H2B, H3, and H4 histone proteins form an octamer, or 8-part structure. What a coincidence! Mr. Histone Man has 8-pack abs. He's basically a nucleosome if you include him with the blue wires. The nucleosome is the basic structural unit of chromatin and contains 2 turns of DNA wrapped around the histone octamer. You'll also see a metal bar that looks like the number 1. That's H1 protein, which is not part of the nucleosome, but holds linker DNA and nucleosomes together. Look, there's more grenades! How fun! These ones are plus charge right next to our histone hostage. Histone proteins have a positive charge because they contain many positively charged arginine and lysine groups, which bind to negatively charged DNA. Chromatin exists in 2 forms, heterochromatin and euchromatin. Thankfully, we've saved several hostages from the nitro gang. Hostage 1 was unfortunately wrapped up very tightly with blue wire. He's got a hat, and like heterochromatin, he's densely packed, not readily accessible, and less likely to be expressed. Poor guy probably can't talk. Heterochromatin looks like a dark blob under an electron microscope, and I'm sure the gang considered him as a dark blob too. Hostage 2 is loosely tied up. He's expressing himself and he's got a U-shirt because he got it on sale, and it also sounds like euchromatin. Euchromatin is less densely packed, readily accessible, and more likely to be expressed. If you look at Hostage 1 again, you'll see a bag of blue meth stuffed in his mouth. I don't know what caused this, but I assume the Nitro Gang didn't want him to talk, so they decided to shut his mouth with a bag of meth. Heterochromatin contains methylated DNA and histones. Just like this bag of meth, methylation condenses chromatin and shuts off gene expression by making it hard for transcription factors to bind and initiate transcription. Also, it rots your teeth and gums. #methmouth. In contrast, Hostage 2 has a vintage cell phone with an acetyl antenna, so he can speak freely. I'm surprised he's even getting any cell phone signal. Histone acetylation removes the positive charge of histone proteins so DNA doesn't bind to them. Acetylation loosens chromatin so transcription factors can bind and initiate transcription. We'll wrap up this sketch by putting a body bag inside the van, because why not? This body bag is technically a bar body, or inactive X chromosome. That's basically when one X chromosome is expressed while the other one isn't. X inactivation, also called lionization, produces bar bodies. They form in genetically normal, 46XX, female somatic cells during early embryogenesis. Males with Klinefelter syndrome, 47XXY, have one bar body, and females with an extra X chromosome, 47XXX, have 2 bar bodies. They look like a compact dark blob near the rim of the nucleus, like heterochromatin. Another example of gene inactivation is imprinting, which occurs when only one gene copy is expressed and the other gene copy is turned off by methylation. Please watch our imprinting video in the clinical genetics section to learn more about the imprinting disorders Angelman and Prader-Willi syndrome. Well, I'm glad our hostages are safe. Let's quickly review what we talked about. DNA contains all genetic information inside cells. DNA is found in the nucleus of eukaryotic cells. Mitochondria also have DNA, but that's circular and passed on by moms. RNA has many forms and is usually single-stranded. Nucleotides consist of a nitrogenous base, deoxyribose or ribose sugar, and a phosphate group. Nucleosides, on the other hand, only have a nitrogenous base and deoxyribose or ribose sugar. DNA has a deoxyribose sugar, while RNA has a ribose sugar. Adenine and guanine are purines, while cytosine, thymine, and uracil are pyrimidines. Uracil replaces thymine in RNA. Phosphodiester bonds hold nucleotides together. Adenine and thymine or uracil are held together by 2 hydrogen bonds, while cytosine and guanine are held together by 3 hydrogen bonds. DNA exists as a double helix and has major and minor grooves. Major grooves form when the backbone is farthest from each other, and minor grooves form when the backbone is closest. Transcription factors and other proteins bind to the major groove. DNA is negatively charged because of the phosphate groups. The nucleosome consists of 2 turns of DNA wrapped around histone proteins, which make an octamer. H1 protein holds linker DNA and nucleosomes together. By the way, histone proteins are positively charged because they have a lot of arginine and lysine groups. That's why they bind so well with negatively charged DNA. DNA and proteins are packaged into chromatin, which condenses during mitosis and exists as heterochromatin or euchromatin. Heterochromatin is densely packed, not readily accessible, and inactive. Euchromatin is loosely packed, easily accessible, and transcriptionally active. Methylation of DNA and histone proteins turn off transcription of genes. In contrast, histone acetylation turns on gene expression by reducing the binding of histone proteins in DNA to allow transcription factors to bind and initiate transcription. Finally, a bar body represents an inactivated X chromosome that usually forms in 46XX female somatic cells during embryogenesis, and is a form of heterochromatin.